discuss digestion of carbohydrates. How carbohydrate is digested. So carbohydrates are different types. We know polysaccharides, oligosaccharides, monosaccharides. Disaccharide, trisaccharides, these are oligosaccharides. Oligosaccharides means the carbohydrates which are having less than 10 units of monosaccharides. So trisaccharide, disaccharides, they are oligosaccharides. Okay? Among the polysaccharides for which we are interested as food, what are these? Starch, glycogen, cellulose. Though we can't digest it, even then we take it because it will serve different purposes. First, it may provide some vitamin C. We are in green leafy vegetables, so we must eat at least 100 grams of green vegetables every day. They will provide some vitamins. They may provide some minerals and fibers. And this undigested part also important because they will form the bulk of the stool. Okay? And if the stool is bulky, then it is easy to evacuate. Otherwise, we feel constipated. So that has got also importance. Among oligosaccharides, so these are we also take dextrine. Among the dextrine is also polysaccharide. But dextrine is what? It is the degraded product of starch, derived product of starch. The molecules number of glucose units, monosaccharides, is much less than starch. The problem is they are having a lot of 1,6-glycosidic linkages, and that makes us difficult to digest them because most of the enzymes that we are dealing with, this one, and from pancreas, these are 1,4-glycosidic linkages, 1,4-glycosidases. So they can break only, hydrolyze only. Breaking means hydrolysis. Hydro means water. With water, we will lyse it. Because when they form the bond, water is evolved. So with that water, we can lyse them. So one hydroxyl group will go to one and hydrogen atom will go to other. In that way, water will be used up again. But 1,6-glycosidic glycosidase are only available in pancreatic juice. In pancreatic juice, right? We see there in pancreatic amylase and dextrinase. Where is dextrine? Here, there, in the succus entericus. So, pancreas, they are not having any dextrinase. See, amyloidic and only pancreatic lipase is there. Pancreatic amylase is there at the bottom. This box, lower box is pancreatic juice, upper one is intestinal juice, and this is the salivary juice. So in the salivary juice only we can see there is only one enzyme, salivary amylase and ptyalin, that can digest carbohydrates. Okay? In stomach, there is no amylolytic enzyme. Means carbohydrate splitting enzymes are not there. Though it is not having, gastric juice are not having any amylolytic enzyme, even then we will see digestion of carbohydrate takes place in stomach to some extent. How? We will discuss. So what are the foods we are having? For carbohydrate we are taking in? And that will contain starch, mainly starch, which are actually will provide us nutrients, starch, and cellulose will not provide any nutrients. Other than this, dextrin we eat, but this is not present usually in nature. While we ferment starch, then as a fermented product, partially fermented product, we can get dextrin. Say in bakery products. In bakery products, they are making bread, biscuits, cakes. So it may have some dextrin as a byproduct of degraded product of starch. And in bakery products, we will get a lot of maltose. We will get a lot of maltose. And these maltose are what? Disaccharides. So among the oligosaccharides, what we eat through our diet? Maltose. Other than this? Common, very common. Every day, yes, who said? Yeah, sucrose. And you are taking some fruits, grapes. They may contain monosaccharides, glucose. So in the nature, we may get glucose. We are eating milk. So milk also contain carbohydrate. What is the name? Lactose. This is also a disaccharide. So what is there in maltose? Glucose, two molecules of glucose. When they are associated with 1,4-glycosidase linkage, they form maltose. In lactose, what is it? It is a disaccharides. Here also we get two monosaccharides. One is glucose, another is galactose. Very nice. And sucrose? Glucose and fructose. So these are the things we actually take through different foods. So our food will contain carbohydrates, and what will be the form? Polysaccharides. For example, starch, mainly the starch. This will constitute nearly 60% of total food. 60% of total food. Among the foods what we are eating, the foods are what? I am debating, coming to nutrition. What are foods? I know what are foods, but I can't tell you what are foods. Is it the situation? Sir? I know what is food, but I can't tell what is food. Foods are substances that are edible. Unless they are edible. Edible means? You intend to eat, you like to eat, you usually eat, you usually take, you usually ingest. So first of all, it should be edible. Otherwise you won't eat. Potato, raw potato, these are having all this, you said. Eat it. Will you eat? Why not? Rice, unboiled rice. We know all these things are present in rice. Will you eat it? Why? It's not edible. The first thing, Sadaji, first thing it should be edible. And usually edible things are having nutrients. Again, what are nutrients? What are nutrients? We eat food because they contain nutrients. Now, what are nutrients? What are nutrients?
Which we get from the food. Which we get from the food. Edible.
What are nutrients? Nutrients are essential. Essential for what?
Growth development.
You are not growing now.
Body weight.
Your growth has been
Stop.
stopped. Then why you are taking food? Nutrients?
Provides immunity.
For the functioning of the body. Functioning of the body.
These are essential for functioning of my body. And we know for functioning, whenever we want to function, do something, function doing something, and this will equal energy. And this energy will be provided by the food. And for this purpose, the food contains something that are nutrients. Okay, you only take food for energy, no? Carbohydrate will not give you protection from diseases. Will it? Will this give? Then you have to take something else for protection of your body. So that's what you need. Vitamins, minerals. Unless you take iron, you will face anemia. Unless you take sodium, potassium, you will lose the excitability of your body. Once it is lost, excitability. See what is happening in ICU patients. We heard, hypokalemia or hyponatremia. Means sodium ion, potassium ion, these are imbalanced, either lowered or it is excess. And it is making lot of problem in the body. Patient may die because of this. That means these are very essential. Calcium ion, these are also very much essential. Without the help of calcium, I cannot contract my muscle. So in that way, we will find lot of minerals. They are essential for our body. They are performing some enzymes. They cannot act alone. They cannot act alone. Some enzymes can act alone. But some needs, hey, without you I can function, come with me. So they need the assistance of others compounds. They are coenzymes or cofactors. So you will find some cofactors, ATPase. Without magnesium it cannot act. Without ATPase you cannot derive energy from ATP. You cannot break down ATP. You have lot of energy, lot of ATP. But if ATPase doesn't act, then it is not possible to break the ATP. You are having lot of money, but you are unable to spend. Lot of money you are having, like Anubrata Mandal. No, not Anubrata. What is his name? No. Education Minister of
WBC.
Still, no. Partha Chatterjee. Yes. A room filled up with currencies. Huh? So, see the consequence. Now he's unable to spend a single penny from there. So, if there is no magnesium ion, ATPase will not be able to act. So, we could understand the necessity of different elements in our body, microelements as well as macroelements. All are needed. So these are present in food. And what these minerals we are having, they will not be staying in my body permanently. They are going out through urine, through stool, through my sweats. So through different secretions, I am losing. So I have to get that, no? So that for purpose we have to eat from external sources through food. Okay? Similarly, you are not though growing, you have a cut. You have a cut. What will happen to this injury? After some days, it will be repaired.
Like new cells.
Which nutrients did it? Who did it? For repairing this purpose, you needed new cells. You needed new cells. So how these new cells are generated? From what? Mainly what? Proteins. For generation of a new cell, we need protoplasm. The protoplasm are mainly proteins. If you see the structure of a cell, every structure is having proteins and lipids. So they are essential. The RNAs, DNAs needed. RNA is having carbohydrates. DNA is having carbohydrates. Can you name them? Say DNA to DNA, RNA to वहाँ से इधर carbohydrate कहाँ से आ गया? तो ribose, deoxyribose, these are carbohydrates. So, to make a new cell, we need fat, we need proteins. Okay, sir, protein is not, आप काम चला लो, मैं fat देता हूँ, lipid you take, carbohydrate you take, can I make the cell? No. Because cell will be performing various functions, and for this functioning, it will be performing various metabolic processes. For these metabolic processes, metabolism means what? What do you mean by metabolism? In chemical reactions. And these chemical reactions are metabolic reactions. Metabolism. Not a single reaction. Numbers of reactions. And some reactions are anabolic. They will produce something. And some are catabolic. They will break these substances which has been produced earlier. So metabolism is maybe either anabolic and catabolic. Now in my body, you tell which process is anabolic process is going on mostly or catabolic process is going on. Yeah, catabolic. When you will be aging, your catabolic process will be more. That's why you will be losing your weight, losing functioning of various cells. Because you can survive for a particular period. Thereafter, you have to die. You have to die. So, not on a sudden, but slowly I will kill you. You will not recover, you will not realize that you are going to die. Slowly, slowly. A baby is growing daily by some, but mother, father, they can't notice it. Every day it was growing, but every day's growth is not noticed. When you smoke, it is deteriorating your body functions, your body. But you will not be able to notice because these are minute, drop, 1 drop, 2 drop, 3 drop, 4 drop. Ultimately, it will be bigger problem. Okay? So, we eat polysaccharides. These are the polysaccharides we eat. These are the oligosaccharides we eat. These are the monosaccharides we eat. So all these foods, having all these carbohydrates, we enter through food in our mouth. Then what we do, we will relish it. When the food is delicious, what we do? We relish it. We'll keep it for longer period of time to relish it. And if it is not, say you are taking, what is this? No, potato, not potato. This is a bit of Kerala. So immediately you swallow it. Immediately you swallow it. You don't relish it. So immediately you are swallowing it. But the delicious food you are. So make the food delicious. These are foods, there are a lot of chemicals actually. So this chemical has to be released. So for releasing these chemicals, there should be some solvent. In this solvent, this chemical should be released. And from this solvent, this substance will be tested by my taste bud. And where from this solvent will come? From the secretions. A lot of water will be secreted. So, this water will dissolve some substances in the food and that will be tasted by my tongue. Taste buds. Then only, yeah, it's good, tasted good. My brain said it's good. So, let us taste it. So, then what you will do? I will masticate it. You will not be keeping just on your tongue or at the side like Hanumanji, one side of your. So, you will be relishing it. You will be masticating it, then you will be swallowing it. So, by this time, what happened? You have the salivary juice also secreted some amylolytic enzyme. What is this? Salivary amylase or alpha salivary amylase. So this is a carbohydrate splitting enzyme. So this will act on polysaccharides, starch, glycogen. Okay. So it will, these are nothing but 1,4-glycosidase, alpha-1,4-glycosidase. So they will break the glycosidic linkages. Glycosidic linkages. You think the starch as a, what is this? Bunch of grapes. These grapes are glucose. Bunch of grapes. You think of each grape as glucose. So there are a lot of glucose connected by branches. So these branches are holding, and in our case, another thing is holding the glucose. There are 1,4-glycosidic linkages. 1,6-glycosidic linkages are actually the branch. Okay, so this enzyme will break this 1,4-glycosidic linkage. As a result, what will happen? The big bunch will be splitted into smaller bunch. Okay? These are oligosaccharides. So then we'll get oligosaccharide. We'll get maltose. Okay, not sucrose. How can it be sucrose? Because starch. What are homoglycans? You are specialist in carbohydrate chemistry, all of you. What are homoglycans? What are heteroglycans? Understood. Homoglycans are those compounds, those carbohydrates. If you hydrolyze them, you will get monosaccharides. All monosaccharides will be same. Starch is a homoglycan. So its hydrolysis will give only glucose. Inulin,
Fructose.
you will get only fructose. No other monosaccharides. So homoglycans, fructan, we'll call it fructan. Glucan or glycan, these are starch, example of glycan. Okay, so from glucan, how can you get fructose? Who said fructose? So from starch you can't get. Starch you can't get fructose, you will get only glucose. So ultimately by the action of salivary amylase, what you will get? Not glucose, never. You won't get any glucose. You will get maltose, isomaltose, maltotriose, dextrine. Okay? These are the products of digestion of salivary amylase. So this will continue its digestion as long as the food is in your mouth. Even it may continue when it is traveling to the stomach. So when it enters into the stomach, its external environment is changed. Earlier it was not acidic. Now it is acidic because of secretion of, it is not there, hydrochloric acid. Is it needed, secretion of hydrochloric acid by the stomach?
Yes, sir.
Okay. So, as long as pH is not lowered below 5 or 6, the activity of salivary amylase will go on in the stomach, particularly inside the food bolus. All these food are masticated here, no? So inside is also the salivary amylase. So outside is getting the food bolus, the outside is getting hydrochloric acid, but inside is not. It will get, inside will also become, will be broken, no? The food bolus will be broken by the crunching mechanism of, by the mechanism of the stomach. Okay, ultimately this will be broken. So when this will be broken, salivary amylase will lose its activity. Now, in this pH, it cannot act. It is deactivated. All the enzymes, one of the particular thing is, they are very sensitive. They are very sensitive, very emotional. You can't say anything. You can't say anything. If any change, they will lose function. They will not, I am going. I will not act. They are sensitive to temperature. They are sensitive to pH. So in a particular temperature, in a particular pH, they act best. And if you decrease the pH, or increase the pH, or increase the temperature, or decrease the temperature, they will lose activity. They will lose their temper. They will not be acting. Okay, so because the change of temperature, the change of pH, amylase, salivary amylase lost their activity. Okay, okay. Oh, I don't need you now. Whatever, how much you have been done, this is okay. Now, stomach. The food is entered into the stomach, and digestion by the salivary amylase completed. Are there any amylolytic enzyme in stomach? No. So I will remain, as long as food is there inside the stomach, as long as it is there inside the stomach, there will not be any more digestion of carbohydrates. You have to wait for some time. When it will be entering to the next part, then only carbohydrate digestion will be starting. So, in stomach, their activity completed, but during this time, something may happen, though there is no any amylolytic enzyme. There is hydrochloric acid. So it can do some job. Okay? I'm giving you one example. In your biochemistry class, you have done a test with sucrose to prove that sucrose is a non-reducing sugar. Benedict test. And so there is no brick-red precipitate formation. When you added Benedict reagent and then heated, so you saw nothing has been changed. Because the carbohydrate, these are not reducing. These are not having any reducing property. That's why you couldn't help. So then what you did? Then you performed another test, hydrolysis cum Benedict test. Okay, I have seen Benedict test, it is not responding, so it is. Then you are adding some hydrochloric acid, then you are heating, neutralizing it with sodium carbonate. Then you are adding again Benedict reagent and heating it. Hurry! What is happening? Break it. Why? Because by hydrochloric acid, you have hydrolyzed the sucrose into glucose and fructose. Now both are reducing. One is having ketone group as reducing group and another is having aldehyde group as reducing group. Now they will reduce the Benedict reagent. That means hydrochloric acid can hydrolyze sucrose. So we have in our stomach hydrochloric acid. So some amount of sugar, sucrose can be hydrolyzed into glucose and fructose. Hydrolysis can be done, though there is no any enzyme there. Is that clear to you? Okay, this much has been done in stomach. Okay, now there is one sphincter in between stomach and intestine. Okay, this sphincter is closed because it doesn't allow too much of acid-rich food chyme. It will not be allowing too much. Otherwise, your duodenum will face Pain, pain, pain. What happened? There is duodenal ulcer. Who did it? This hydrochloric acid. This hydrochloric acid did this. And who is the responsible person? This sphincter. Why you have allowed this? With so much of acidity, so much of chyme, why you have allowed? This acid, hydrochloric acid, actually destroyed the intestinal epithelium, duodenal epithelium. So you do your duty perfectly, sphincter. You do your duty perfectly. So you send the sphincter, you send a bolus, a chyme. So here it is the duodenum. The duodenal wall will feel, this is acidic. So immediately it has to be neutralized. So a lot of bicarbonate ion will be secreted by its own wall, as well as some secretions will be there. These secretions are hormones. They will come to, these hormones will cause pancreas to release bicarbonate-rich secretions. So these secretions and the secretions of bile from gallbladder, they are also having a lot of bicarbonate ions. So all this bicarbonate ion will start to neutralize this food bolus. Okay, this is neutralized, go. Environment is now alkaline, not acidic, go. Next bolus come, neutralize it, go. Next bolus come, go. So this way it will be allowed. When it is so much of acidic, the chyme is so much of acidic, all the chyme present in our stomach lumen, so acidic, it has gone beyond the capacity of the duodenum to neutralize. So sphincter will close. So as a result, there will be stagnance. What is it? Indigestion. Neither it is allowed to go to the next part, and it is creating a lot of problem in your stomach. Then discomfort, nausea, vomiting. So with this vomiting, this food product will be coming into your esophagus. It may damage in that case. It happens repeatedly. It will also damage. It will cause ulceration in the esophagus. You will feel, so acidic. So its pH may be 2. So much acidic it is. Okay, so in that way, so whenever you take food, so you must relish it first. You should not do hurry, class shuru ho jayega, jaldi finish karo lunch. Then take less. Better than you take less. But it should not create any problem there. You don't take so much of spicy food. Some foods are very irritating to the stomach. Some foods will release so much of hydrochloric acid. So that will harm your body. So you should not take. It will invite, hey, hello, come, eat me. My smell is very good, no? So come, take this. God has given you some time period. You will be, if you follow my direction, my direction, nature. Nature has given you, nature has produced you and given you something in your body to survive in the nature. But what you are doing, you are not abiding by the law of nature. You are not using what you are having. Okay, you are not using it. See, the duck, the hen, they have their wing, but they cannot fly. Why? Why they cannot fly?
They don't know how to fly.
But they knew.
It's not wings.
We, the human being, actually we made them like this. When we found that it lay eggs, and these eggs are good food to me, and its flesh is very nice to eat. So let us make it a domestic one. So these birds or these animals no need to collect their own food. My owner will be providing my food. And if somebody comes to kill me, my owner will protect me. So for protection, why I need flying? Either for protection or to collect food. So everything is supplied by the owner. So why should I fly? So I made them. We made them like that. See, some hen are there in forest. They can fly. Because they had to collect their own food and they had to protect themselves. So their wings are very activated. So we made them to adapt themselves in our situation. Evolution made. Evolution is made through adaptations, gradual adaptations. You lost your through adaptations, gradual adaptations. Okay. The food entered in, neutralized, entered in duodenal wall. Now what will be there? There are juice, some carbohydrate splitting juice, amylolytic enzymes are there. First, some amylolytic enzymes will get from pancreas and some will be through intestinal juice. So let us see what is there in pancreatic juice. Amylolytic enzyme, pancreatic amylase. It will act like salivary amylase. Only difference is the environment, pH environment. And another difference is that pancreatic amylase. So what you did today, there is this much of boiled rice is present, but I am feeling very hungry. So unboiled rice, let us eat it. So you ate. So that one not digested, that unboiled rice were not digested by the salivary juice. These will be digested by. So this can act on unboiled polysaccharide, starch. Okay? So they can digest the glycogen. We call the glycogen, you will be, if you are eating mutton, and your mutton may contain muscles, and definitely they will be there, invariably they will be there. Muscles will be there, and muscle can also deposit some glucose. Glycogen. So when this will be acting, so that glycogen will be broken down and utilized for energy. Liver also deposits some glucose as glycogen. So if you take liver, so liver will be having glycogen. Glycogen is starch, like animal starch. We call this glycogen animal. Can you name one animal starch? Yes, glycogen is the animal starch. These are also having 1,4 glycosidic linkase, 1,6 glycosidic linkase. This will also produce dextrin. When it will be degraded, it will also produce dextrin. So as a result, the amylolytic enzyme will also act on polysaccharides and produce a lot of degraded products. Now what will happen to these degraded products? Dextrin disaccharides, triglycerides. So to digest them, there are enzymes in succus and entericus. What are these? Amylolytic enzymes, sucrase, maltase, lactase, dextrinase, trehalase. So what this sucrase will do? In stomach, what has been done by the hydrochloric acid?
Breaking down
Of sucrose. So here enzyme we are having sucrase. So rest of the sucrose will be digested by, hydrolyzed by this, and we will get fructose and glucose. And there is also, I can see what there is in succus entericus, there is maltase. We know the maltose are the product of starch because of digestion by the amylases. Pancreatic amylase and salivary amylase, they have produced maltose. Now this maltose will be digested by the succus entericus maltase, and they will produce 2-glucose. Similarly, in the intestinal lumen you will get lactose. You have taken milk, no? So it has to be digested. So this lactase will digest this lactose into glucose and fructose. And there is dextrinase, lot of dextrin, having lot of 1,6-glycosidase enzyme. Now dextrinase are actually 1,6-glycosidase. So they will break all the 1,6-glycosidic bonds. As a result, now it is very easy for the 1,4-glycosidase, salivary, pancreatic amylase to split all those glucose from them. So in this way, and what are other enzyme? So we got the action of, we know what the function of sucrase, functions of maltase, lactase, and dextrinase, and last one, trehalase. It acts on trehalose. What is this? Trehalose. These are actually disaccharide. These are also like maltose made of glucose and glucose. 2 glucose units are there, but they are present in some special plants, special animals, insects. We love eating insects. If we means we are Chinese people. We like, cockroach, fine. Yes, Thailand. Bangkok.
Thailand they eat a lot.
Lot, no?
Any streets who went
So these insects, no? They need to fly for a long period of time. They can cross sea also. Some flies, no? Butterfly, they can cross the sea also. So when they fly this much of distance, then where the flying means they will be needing energy. They will be needing water. To where from they will get the water, where from they will get the energy? See, can you eat the seawater? They are also not able to drink. Okay, so what is there? In this trehalose molecule, there are water. It is a lot of water there within this molecule. So this molecule they use and this glucose they use for energy. So the scarcity for water and for energy is made by the trehalose. In desert, the plants will also having this side of this. There is scarcity of water. So this molecule may provide hydration to them. So that's why these are important, trehalose. But if we eat those plants or these insects, it's okay. Then it has to be digested, no? That's okay. You may go to China or go to Bangkok. So there you may taste insects, okay? So then you have to digest this trehalose, no? So that way it is very important. And Hopefully, my carbohydrate digestion is completed by this time. The hematological indices, there are other hematological indices that we will have to calculate. Say, during your exam, we have given one question. You calculate the mean corpuscular volume, MCV, of your patient, of your subject. So, subject is given to you. Now, what you will do? What you need? That much, we will tell that much. And you determine or you make inference. Is it normal or abnormal? What happens if it increases, it decreases? You write that. So, what you will do? I have already mentioned, determine MCV. So, what you will do? You need to do two experiments. One is RBC count of this blood. Another is RBC count. Another is hemoglobin concentration. Not hemoglobin. Packed cell volume. PCB. Packed cell volume means what is the packed cell? You have to know. First you determine the packed cell volume. Then you determine the RBC count. Then only you will be able to calculate this. So in this way, when the MCSC will be given, so you know what experiments you have to do first. Then only you can determine. Or it can be done also. We will give one value of one, say value of any one experiment can be given to you. Then you have to determine the other, and then you have to determine this. So let's see what can be done. First you have to do both the experiments. Then you will be determining.